Hello everyone. Welcome back to PSN Bio Academy. If you are new to my channel, this is Vazna Imran, and this is the place where you can top up your biology knowledge with our short and clear past paper videos. Look at the past paper questions. Based on those questions, I have made some categories. They are chemistry of life, mammalian transport system, cardiovascular health and risk, membrane and transport, proteins and DNA, and gene expression and genetics. So right now we are still under chemistry of life, and under chemistry of life, these are the subtopics I have already made. Today we are going to tackle some questions on lipids. So lipids, when I bring up this topic, lipids, their structures are carbon rich and hydrogen rich. Think about things like cheese or oil, and then you can also see the structure of cell membrane. Importantly, when you think about lipids, you also have to remember things like chest pain, especially heart attacks or cardiovascular risk. But what you should know about lipids as an IAL student is what's most important here. So basically, I can divide lipids into four categories according to your past paper. They are triglyceride, phospholipids, cholesterol, and lipoproteins. So these are the terms you need to know to answer today's questions. Now, again, under triglycerides, there are two parts which make a triglyceride. One is glycerol molecule, and you know glycerol, this is how it should be. It's an alcohol group. So in organic chemistry, you might learn that alcohols have an OH group. Glycerol actually has three alcohol groups, so three OH groups, as you can see. And the other part of a triglyceride is fatty acids. So both glycerol and fatty acids together make a triglyceride. Now, looking at fatty acids again, the acid part, you might remember from organic chemistry that acids have a COOH group. Here, fatty acids also carry one COOH group. In fatty acids, again, we have two types. The one is saturated and the other one is unsaturated. In saturated fatty acids, the number of carbons can differ, but they are saturated with hydrogen. So that means they don't have any double bonds in the fatty acid chain. But compared to the unsaturated fatty acids, so they are also acids. So they have a COOH group, but they are unsaturated. So this means they are missing some hydrogen, or they can add with some hydrogen atoms more. So they usually have one or more double bonds in their tail part. Now let's look at how a triglyceride molecule is formed. One glycerol and three fatty acids join together to form a triglyceride through a condensation reaction. A condensation reaction means water molecules are removed and a bond is formed called an ester bond. This is what you can see in your textbook. And keep remember, in past papers, they have already asked you to draw other bonds like glycosidic bonds and peptide bonds. But this one is still possible. So maybe in the future, they can ask this question as well. So keep in mind that you have to draw three ester bonds. So now if we move to phospholipids, again, there are three parts that make a phospholipid: one glycerol, two fatty acids, and one phosphate group. Actually, phospholipids also similar to triglyceride, but what has changed is that instead of one fatty acid chain, it's replaced by a phosphate group. Because of this, they have special properties, a hydrophilic head and hydrophobic tails. And this is the structure of a phospholipid. You can see that the phosphate groups gives a hydrophilic head-like structure, while the tails give a hydrophobic property. So that's all you need to remember about phospholipids. And the next part is cholesterol. So here I'm not going to talk about cholesterol. We'll learn about cholesterol when we study the cell membrane structure. So let's skip for now and move on to lipoproteins. So both cholesterol and lipoproteins are very important when you are learning about cardiovascular health and risk. Anyway, you should know now what a lipoprotein is here. So again, lipoproteins, lipids, and proteins. From the name itself, we can understand that they are made up of lipids and proteins, and the special proteins called apoproteins. So what do lipoproteins do? They are carriers. So they carry cholesterol inside the body, either from the liver to body cells or from body cells back to the liver. So that's how they transferred it. If you look lipoproteins, again, we can divide them into HDL and LDL. So HDL means high density lipoproteins and LDL means low density lipoproteins. And this structure here actually represents a lipoprotein, the apoprotein part, phospholipid, and blue indicates cholesterol and yellow is triacyl glycerol. And you can see that HDL has a higher protein content compared to the LDL. And if you compare both, LDL have more cholesterol and less proteins, while HDL have more protein and less cholesterol. Right now, let's jump into the question part, which is the most important. And the first question is coming from the October 2020 paper, question number 8. So you have a small description here. If you want, you can pause and read it. Okay, it's simple and clear. Here you can see the basic parts of the lipoprotein. They have used a representative diagram, not an actual image, so don't worry too much about the picture. The outer layer is phospholipid and proteins. Then inside you have triglycerides and the core part is cholesterol. And the question says compare and contrast the structure of a triglyceride and a phospholipid. Whenever you see compare and contrast, you know you have to write both similarities and differences. If you write only similarities or only differences, you will lose marks, so you must include both. From what we have already learned, the structure of both triglyceride and phospholipids are quite simple. And now you can write for similarities. Both triglycerides and phospholipids contain glycerol and fatty acids. They also both have ester bonds, so you need to mention that. And for differences, triglycerides have three fatty acid chains, and whereas phospholipids have only two fatty acid chains. So the number of fatty acids is different. Also, phospholipid contain a phosphate group which is very special, but triglycerides do not. You can write your answer in a table using bullet points or in paragraph form. Personally, I prefer paragraph form because it helps you answer questions more confidently. For example, both triglycerides and phospholipids contain glycerol and fatty acids. However, triglycerides have three fatty acid chains, while phospholipids contain only two fatty acid chains. In addition, phospholipids have a phosphate group, whereas triglycerides do not. And both molecules are linked by ester bonds. If you find a table easier, that's totally fine. But I encourage my students to practice writing in paragraph form because in many other questions like explain or discuss, you will need to write paragraph anyway. And the next question is explain how the properties of LDLs enable cholesterol to be transported in the blood. This question is a bit more logical. You have to construct your answer more logically than just theoretically. So you need to think what features help LDLs carry cholesterol. This is the structure they have given phospholipid and protein on the outside, and triglyceride and cholesterol on the inside. First, remember that lipoproteins are carriers. The question itself mentioned that they are carriers, right? So they must be able to dissolve in blood plasma. This is the first thing you need to remember. Blood plasma is hydrophilic since it contains water. If you look at the outer part of LDL, it has phospholipids and proteins. The phospholipid heads are hydrophilic and proteins are also hydrophilic. So this allows LDL to dissolve and move in the blood plasma. And this picture is important because even though they only show phospholipids, you have to think beyond the diagram. This idea has been tested many times in past papers. Phospholipids can arrange themselves according to the environment. They are magicals, right? The hydrophilic heads face towards the blood, while the hydrophobic tails face inwards. At the same time, LDL has to carry cholesterol, which is hydrophobic and doesn't dissolve in water. So, inside the LDL, cholesterol is surrounded by the hydrophobic fatty acid tails of phospholipids and triglycerides. This creates a hydrophobic interior that can hold cholesterol safely. So, these are the two main ideas you need to explain in your answer. You can write in your own language, but make sure you include these key points because these are the marking points that examiner are looking for.
So the similar type of question we can see again in 2024 Jan paper. Here also you are given with a diagram and this time it's about a lipid nanoparticles, but the concept is same. The question asks, explain the arrangement of phospholipid in the nanoparticles that carries mRNA. If you look at the structure clearly, first you have plasma, it's hydrophilic, so definitely the phospholipid head should be faced to the hydrophilic environment. Then here it's something little bit advanced than the previous question. The mRNA has been surrounded by phospholipid heads within the particle because mRNA is polar and they cannot tolerate the tail part. So finally you can see all these hydrophobic tails have been facing each other as all of them are hydrophobic. So you have to include these points in your answer. For example, you can write the outer layer of lipid nanoparticles is made up of phospholipid heads, which are hydrophilic, so they can interact with the plasma or aqueous environment. The mRNA inside the lipid nanoparticles is surrounded by phospholipid heads because it's polar. The hydrophobic tails of the phospholipids in the outer layer and those surrounding the mRNA face each other as they are hydrophobic.
Now let's continue with the 2020 paper, and this part is a simple calculation question. You are asked to complete the table by calculating the volume of LDL and the ratio of LDL volume to cholesterol volume. First, let's calculate the volume of LDL. Here you are given the diameter as 24. Please remember, when calculating volume, we must use the radius, not the diameter. So if the diameter is 24, the radius is 12. Now we use the formula for the volume of a sphere, 4 by 3 pi r cubed. So this becomes 4 by 3 pi multiplied by 12 cubed. So when you calculate this, the volume of LDL comes out to be 7,238. Next, let's calculate the ratio. The cholesterol volume given is 523, so the ratio is 7,238 divided by 523. It gives a value of 13.8. In exams, it's better to give the ratio as a whole number, so we round 13.8 to the nearest whole number, which is 14. So the final answers are 7,238 and 14 is to 1.
And the next question is coming from the June 2021 paper. They ask how an unsaturated triglyceride is synthesized. You have to focus on two words, unsaturated and synthesized. Triglycerides are formed from one glycerol and three fatty acids by a condensation reaction. Since the question says synthesized, mention that the body makes it using specific enzymes. And because it's unsaturated, at least one fatty acid has one or more double bonds.
Now let's compare this with another question from the 2023 January paper where they simply ask you to describe the structure of a triglyceride. Here you don't need to write anything about synthesis or enzymes. So you just state that a triglyceride consists of one glycerol molecule joined to three fatty acids by ester bonds. That's all.
The next question is from October 2021, paper 4A. This is a very common and well known past paper question, so it's important to understand it clearly. In this question, they are asking about a saturated lipid. Please keep in mind that the focus is on saturated fatty acids. They are asking about carbon carbon double bonds and the carbon is to hydrogen ratio. First, let's review what a saturated fatty acid is. A saturated fatty acid doesn't contain any carbon carbon double bonds. Please note, this doesn't mean there are no double bonds at all. Saturated fatty acids still contain carbon is to oxygen double bonds as part of the carboxyl group, but they don't have carbon to carbon double bonds along the hydrocarbon chain. And the general formula for a saturated fatty acid is CnH2n plus 2. Now, let's compare this with unsaturated fatty acids. So, unsaturated fatty acids contain one or more carbon to carbon double bonds. If there is one double bond, the formula becomes CnH2n. So this difference affects the number of hydrogen atoms attached to the carbon chain. So let's look at an example to clear up a common confusion that students have. Assume both fatty acids contain 18 carbon atoms. And for saturated fatty acids, the formula will be carbon 18 times hydrogen 38. So the carbon is to hydrogen ratio should be 18 is to 38. And for unsaturated fatty acid with one double bond, the formula can be written C 18 times and hydrogen 36 times. So the ratio should be 18 is to 36. Now, if you calculate the ratios numerically, I want to prove that mathematically. So, for saturated, it should be like approximately like 0.47. And for unsaturated, it is solid, it's 0.5. So, the carbon is to hydrogen ratio is higher in unsaturated fatty acids than in saturated fatty acids. So final answer is answer B. Carbon carbon double bond is absent and carbon is to hydrogen ratio is lower than in an unsaturated fatty acid chain. The next question is from the June 2022 paper. It's a similar question but it tests a different concept. And this time the question is about an unsaturated fatty acid. Unsaturated fatty acids do contain carbon-carbon double bonds. It's obvious. Because of these double bonds, unsaturated fatty acids have fewer hydrogen atoms compared to saturated fatty acids with the same number of carbon atoms. And this question focuses on the number of hydrogen, not the carbon is to hydrogen ratio. So the answer, yes and fewer. Another question is from the May 2023 paper. This question is very similar to the October one, but this time it focuses on an unsaturated fatty acid. Unsaturated fatty acids do contain carbon to carbon double bonds, and when compared with saturated fatty acids that have the same number of carbon atoms, Unsaturated fatty acids have a higher carbon to hydrogen ratio. This has already been shown numerically. So answer is very simple. Yes. And then they have higher than the saturated one. Now let's go back to the 2021 question, but this is a different question. Two diagrams are given, HDL and altered HDL. HDL functions, they are cholesterol transporter, and the altered HDL carries a higher amount of cholesterol than the normal HDL. And the question asks you to compare and contrast the structures, so you must write similarities and differences based only on the diagrams. So, as you saw, we can write the similarities. Both of them are made up of phospholipid layer and both contain apoprotein A1. And if we look into the differences, we can see altered HDL is look bigger, they larger than the normal HDL. An altered HDL contains more cholesterol esters, CE. You don't need to write the exact word, just write they contain more CE. And if you look closely the picture, and they contain less polyunsaturated PC, the long chain polyunsaturated PC. And if you look that side, the normal HDL, they contains more types of proteins besides proteins A1. So this is a look and write question, not a theoretical one. So like exactly what we did in our small classes. Write as many clear similarities and differences as shown in the diagram to score full marks. The next question is from the June 2022 paper. This is a very simple drawing question. Biology is pretty funny. Sometimes you need to draw, sometimes you need to do some calculation, and most of the time you need to write paragraphs. So here you are asked to draw a phospholipid molecule using the given shape. So it's so simple to draw it. First, let's draw the phosphate group as a circle and attach it to a glycerol molecule represented by a rectangle and the colored rectangle. Then you have to draw two fatty acid chains attached to glycerol, and then you have to show the ester bonds in correct places. So this is a standard phospholipid structure which is commonly drawn in class. It looks like a man which we draw in our small classes. And the same question was repeated in October 2022. So the answer is exactly the same. So let's get back to the question from June 2022, paper part B2. You are given a diagram of a triglyceride molecule and asked to identify the ester bond. A triglyceride contains three ester bonds formed between glycerol and the three fatty acids. So from the diagram, the correct answer is C. The next question is from 2022 October paper, which is very straightforward. Which type of bond is found in this molecule? It's ester bond. Here we go with a question from 2023 January paper, which is very straightforward, which describes a phospholipid. It's obvious they have a hydrophilic phosphate head and hydrophobic fatty acid tails. So answer is B. Now let's go to the question from the January 2024 paper, question number 8A part. Here you are given small piece of information, and finally the diagram shows a part of an unsaturated lipid. Keep in mind that when they talk about an unsaturated lipid, it usually refers to the fatty acid chain, which is the tail part. And this fatty acid chain is unsaturated, meaning it contains one or more carbon-carbon double bonds. Be specific, these are carbon-carbon double bonds. You are asked to complete the diagram to show a possible structure of an unsaturated lipid. So remember, unsaturated means there must be a carbon-carbon double bond. You can add the carbon-carbon double bond in any suitable position along the chain. After that, you need to add the hydrogen atoms. Keep in mind that each carbon atom must form 4 bonds in total. According to this rule, you should add the correct number of hydrogen atoms to each carbon. And next, moving on to another question from the October 2024 paper. Again, question number 8. Apparently, the examiner seems very interested in putting lipid questions as the final question number 8. I hope so. So again, here you start the question with a nice avocado picture, and the question asks you to compare and contrast the structure of an unsaturated fatty acid and a saturated fatty acid. So far in past paper questions, we were usually asked about saturated and unsaturated fatty acids separately. But now it's a recap question where you have to use your knowledge from past paper and write the similarities between them and the differences between. So for similarities, you can write it in a comparison style. On the left side, you have saturated fatty acids, and on the right side you have unsaturated fatty acids. So for similarities you can write both have or both are made up of carbon, hydrogen and oxygen, and both are made up of covalent bonds. But be very careful and be specific. We are talking about fatty acids, not triglycerides. Many students get confused and write that they have ester bonds, but remember there are no ester bonds in fatty acid molecules. Ester bonds are only found in triglycerides, not here. So only write covalent bonds. Now let's come to the differences. Here I'm comparing both, but I'm writing points specific to one molecule only, not writing long sentences like this one has this and the other one has that. Just write the clear points. So for unsaturated fatty acids, they have carbon-carbon double bonds. Be specific. If you write only double bonds, it's wrong because double bonds can also refer to carbon oxygen double bonds, which are already present. The scheme itself makes that clear. Unsaturated fatty acids have fewer hydrogen atoms for the same number of carbon atoms, or you can write they have a higher carbon is to hydrogen ratio. And finally, unsaturated fatty acids are bent because of the presence of the carbon-carbon double bond. The chain is not straight and it becomes bent. So finally, we have come to the end of this session. The latest lipid question is from the Jan 2025 paper. After that, in May and October 25, there were no questions on lipids. So in 2026, we might get more. And this question about the cell membrane, and a part of this question asks about phospholipid. That's why I have brought here. It asks, give 2 ways in which phospholipids can be different from each other. If you look into the diagram carefully, the first way, it can be differ by the fatty acid tails. So one or both tails can be saturated or unsaturated. And also they can be differ by the length of the fatty acid chain. So some phospholipids may have long tails and others can have short tails. And if you look again, they can be differ in the number of double bonds, even though they could have both unsaturated fatty acids. But again, the number of double bonds they are carrying can be differ. And finally, if you look further, the phosphate group in some phospholipids can be attached to different groups like carbohydrates or proteins. So these are the possible ways how two phospholipids can be different from each other.
I hope you found this video useful. Thank you for watching. Please subscribe to PSN Bioacademy, and if you want any past paper questions or from any specific areas, please comment below. Don't forget to subscribe and share with your friends.